Thank you. So in 2014 and 2015, I was able to do some research looking at narrow buffer effectiveness. So what is a narrow buffer? So these are native planted vegetation along streams and agricultural drainage ditches. They're planted there and where there was none before. And along these drainage ditches in the agricultural region, I measured the effectiveness of the buffers by looking at water temperature change from an upstream to a downstream portion of the reach, and I looked at fish populations. I used the fish populations as a response variable to the buffer width that was a treatment for the experiment. So basically, this whole symposium is about reconciling the needs of people and the environment. So buffers kind of are that middle solution to land use, such as farming, and to making the aquatic environment a little bit better. So basic fish ecology in the Pacific Northwest, you know that fish need clear water. Buffers filter out sediment, nutrient inputs to keep it clean, and then they shade it to keep it cold. But farmers' needs also need to be reconciled, and that's where these narrow buffer comes in. So the buffers still need to be able to allow drainage so farmers can use their fields. They can't be too big or else a lot of land is taken out of production, and that leads to lower profits. And what happens if farms can't profit is that we sell them to development, and then we have concrete instead of these green spaces. So this is an important part of our county that we need to think about. But there's also benefits to farmers from planting buffer. They shade the stream, preventing reed canary grass from growing. They create a refuge for native pollinators and pest predators. And then they also create these windbreaks to reduce the loss of their valuable topsoil. I wanted to know to what extent are narrow buffers benefiting the environment? So we know that shade is important, and that shade closest to the stream edge blocks the most solar radiation that has the potential to warm that water. We know that narrow buffers do a great job at shading the stream. We have theoretical evidence. DeWall in 2010 found that a 6 to 7 meter buffer could achieve 80% shade. As far as water temperature, we know that a lot of the research that's out there is for upland, forested areas that we've extrapolated to how we think that will influence water temperature in these lowland areas. And one of the key things to point out is that we know that longer buffers may be more important to patchy, shorter buffers. Because Ryan et al. In 2013 reported that over a 300-meter length of stream, the stream actually cooled 1 whole degree Celsius, which is a lot for 300-meter length of stream. And then for salmon and trout, we know that buffers create some habitat. They're shading the stream. But as far as relative abundance of fish, I couldn't find much research on that. There was one other study in Ontario, Canada, that looked at fish relative abundance in agricultural drainages with buffers and then reference sites. And what they found was that there was no difference between the two populations in these ag buffers. So either those fish populations were already really degraded at both places, or the buffers were doing a great job in matching the habitat in those reference sites. So I wanted to find out what is going on in Whatcom County. So I looked at 15-foot buffers and 35-foot buffers and compared those to sites without buffer. I got these widths because they're within the Conservation Reserve Enhancement Program, which is an incentive program for private landowners to establish buffers. The funding is most often associated with widths of 35 feet or more. And then there's also an option within the CREP program for a 15-foot hedgerow, which is the photo on the left. And then here we have a ditch or a agricultural drainage without any buffer. And as you can see, somebody has sprayed some herbicide here to keep that ditch clean. So my temperature predictions were that, obviously I thought since these buffers are doing a great job of shading the stream, over the course of the 100-meter reach, I thought, well, surely these water temperatures will be maintained, or maybe even because there's this shade, other processes that are happening will allow that water to cool down. Maybe there's some cool groundwater input. And then I thought sites without buffer would obviously warm. My study sites were in Whatcom County, all within the lower Nooksack drainage basin. So I had 14 sites total. It was an unbalanced design. Unfortunately, some of the sites had to be excluded. All of this research was done over a 100-meter reach on sites with the same land use type, which was dairy farms. And I think one of the sites was a berry field. But they were all fish-bearing, and all of the buffered sites were at least 5 years old. And a lot of these were deciduous. Some of them were mixed deciduous and coniferous plantings. So those deciduous trees grow pretty fast. So 5 years or more is plenty enough time to create a good canopy for shading. And then I placed temperature loggers in the upstream and downstream ends of the reach and in the center of the reach on the bank within the shaded portion of the buffer. So there's a lot of things that can influence water temperature. I tried to account for most of these. The only thing I wasn't able to measure was groundwater, which could be important, but I thought, well, I'm doing my study in the warmest part of the year. Maybe it's not as influential during the low flow period. And because shade is one of the things that influences water temperature by influencing the air temperature, I measured the effective shade, which is the percent of shade over the waterway. These photos are examples of 4% shade, 48% shade, 81% shade, and 90% shade. And there are a series of photos along the buffered reach that are averaged to get the average effective shade for each site. And then I also looked at vegetation height, species density, and the type of species. And when I sampled the fish populations, I looked at the relative abundance, so the number of individual species compared to the total number of species at each site. I did this using the electrofisher. I just did one pass. So as you can see, the results for the shade were that in 2014 and 2015 on the right, the levels of shade for the zero or no buffered sites on the left, 15-foot buffer in the center here, and 35-foot buffer on the right. In both years, the buffered sites were pretty similar, so I could assume that shade was influencing air temperature and water temperature similarly. And then I tested the influence of water temperature by looking at temperatures above 17.5 degrees Celsius. What was at those temperatures, were the streams continuing to warm? Were they cooling a little bit? And the reason I chose 17.5 degrees Celsius as a base temperature to compute the differences in upstream and downstream were that is the US EPA criteria for migration and rearing stream temperature. So, and you can see that here's a little reminder of how I calculated the daily values of the difference between the upstream and the downstream temperatures. I accumulated the differences over a day, and then I averaged those over a week. Several of the streams, this data is only from 2015 because there was one site in 2014, a much cooler year, that there's only one site that went above 17.5 degrees. So, which is good for fish, bad for master's thesis. But, so 2015, I was only limited to 2 sites with 0 buffer, 2 sites for 15-foot buffer, and 1 site for the 35-foot buffer. So I wanted to compare between sites, but to have a meaningful statistical analysis, the sample size was too small. So what you see here is, here's our 0 line here. And so the difference in temperature at temperatures above 17 degrees are only within 1/10 of a degree. So the difference is pretty small from the upstream to downstream. But you can see for sites without a buffer, most of these points throughout the season, on average, the difference in temperature was increasing. So that means at temperatures above 17.5 degrees C, the water continued to warm. And they follow similar patterns at the 0 and the 15-foot buffer site. But as for the 35-foot buffer site, it's showing something different, where the average weekly difference was almost 2/10 a degree cooler over that reach. So I did some other analyses to kind of figure out why I got those mixed results, but I'm going to skip along to my fish analysis. So I did compare relative abundance between sites for fish populations, and I used an analysis called multidimensional scaling. And the first rule of multidimensional scaling is that there is no scale. So how you interpret this plot is by looking at the relative difference between points. Each one of these points is a site, and I've labeled them by width. If we're looking for similarity in the groupings of sites, so the results I have here are that there were no differences in any of the populations of fish for either year, for 2014 or 2015. If we were to see differences, you might have a grouping of 15-foot sites in one location, and then a grouping of 0 sites in another location. And the distance between them would be great enough that would be a statistically significant distance away from one another. But instead, we see this random pattern that it just means that the fish populations, there wasn't anything different happening within those fish populations according to buffer width. So that's kind of what the study in Ontario, Canada saw. So that's something that's interesting, even though I was really hoping that the 15 and 35-foot buffer would show a difference. So, and here's just some fish pictures. I'll let you read my summary.
I was interested in your lack of fish response detection with that, and curious if you thought that one potential reason for that is either that the buffers that have been planted, either the composition of those buffers or not enough time to where you've got other factors happening, like either small or large, litter going into the stream, or wood, where you get habitat unit development and things like that that haven't yet happened. I would expect some response if the right trees are planted with to get habitat formation as well as shading. Mm-hmm. And I just was curious if you had any thoughts on that.
So I did a pebble count for substrate, and I also looked at, there's quite a few things that I measured that I didn't have time to explain. So I did look at that. These sites were agricultural drainages. They're really incised. A lot of them were pretty narrow, less than 5 meters wide. Most of them 2 and 3 meters. Not a whole lot of pools or large woody, almost no large woody debris. You have to remember these drainages are used to drain, so it's really hard to get complex habitat in these areas.
Actually, it's a really nice thing you're studying this. It'd be nice if you could continue and get a larger sample size. The species diversity itself can have a significant element, as you had, I think, said earlier, but that shouldn't be underestimated. And then another thing is that when you have a buffer there, we can't assume that the effects ecologically coincide with the exact geometry. Mm-hmm. And that's something to be kept in mind.
Mm-hmm. Yeah. I happen to know that there is money with the King County Conservation District to fund some buffer research, and been in contact with them. So I think they might be going forward with a larger scale study where they are in ag lands. So hopefully they get some more interesting results and carry this work on.
Great. Thank you, Jessica.